Hello guys, I'm Shifan, and in this video we'll be going through the next 20 set of MCQs from the 2023 biology MCQ paper. Now on our last video we went through the first 20 MCQs from the 2023 paper, and the link for that will be given below. Now let's go through question 21. Which of the following indicates the forms that transport the lowest and highest percentage of carbon dioxide in the human blood? We know as humans, we inhale oxygen and exhale carbon dioxide. Now, where is this carbon dioxide coming from? This carbon dioxide is coming from the tissues. From the tissues, they diffuse into the blood. And through the blood, they are transported into the capillaries in the lungs. Then from the capillaries in the lungs, they get transported to the alveoli, and from the alveoli, they get exhaled out of the lungs. So this is the overall view of how carbon dioxide exhalation takes place. Now, we know carbon dioxide is transported in the blood, but carbon dioxide is not mainly transported as its original form, which is carbon dioxide. It is not transported that way, but it is transported mainly in two other ways. So, first way is has bicarbonate, and second way has carboaminohemoglobin. And also a very small amount as pure carbon dioxide is transported in the blood. Now, which is the highest percentage? So, highest percentage of carbon dioxide is transported as bicarbonate. How does this happen? Now, we know carbon dioxide is present in the tissues, right? From the tissues, they diffuse into the blood. And in the blood, the carbon dioxide enter into the red blood cells. Inside the red blood cells, using specific enzymes, carbon dioxide binds with water to form carbonic acid. Now this carbonic acid dissociates into bicarbonate and hydrogen ions. This bicarbonate, now that is present inside the red blood cells, they come out of the red blood cell and is transported through the blood into the capillaries in the lungs, where again the bicarbonate that is present goes into the inside the red blood cells and become carbon dioxide and comes out and goes into the alveoli. Now this is how transportation as bicarbonate occurs, and this is the highest form. And the lowest form occurs as dissolved carbon dioxide, which means carbon dioxide doesn't bind or react with anything. Carbon dioxide, when it comes out of the tissues into the blood, has carbon dioxide, it is transported through the blood. So that's what dissolved carbon dioxide is, and that is the lowest form. Above, on average, it's like 7% of carbon dioxide is transported as dissolved CO2. And the correct answer is the fifth one, because highest percentage is CO3 and the lowest percentage is dissolved carbon dioxide. Now, okay, I think I can highlight it, sorry. Okay, now that we have seen the answer, there's one more thing I just wanted to teach you guys. It's about carboaminohemoglobin. If you haven't learned it about carboaminohemoglobin, so carboaminohemoglobin is formed when carbon dioxide that is present in the blood, right? As I told, the carbon dioxide becomes bicarbonate, and some carbon dioxide doesn't react with anything. They are transported as pure carbon dioxide, but there's a portion of carbon dioxide. What they do is they go and bind to the hemoglobin. Now, they don't bind to the part where oxygen binds. They go and bind to another part of the hemoglobin, specifically to the amino acid part, amino group of the hemoglobin. Now, they bind to the amino group of the hemoglobin and form carboaminohemoglobin, and carbon dioxide is transported with the red blood cells. Right? So, that's what carboaminohemoglobin is, and 20% of carbon dioxide is transported in this way. Okay. Now let's go through question 22, sorry. If the tidal volume, residual volume and inspirated reserve volume and expirated reserve volume of a person are 500 milliliters, 1200 milliliters, 3100 milliliters and 1100 milliliters respectively, vital capacity of this person is. Okay. We know for the vital capacity we have an equation, right? So the equation is tidal volume plus inspirated reserve volume plus the expiratory reserve volume. So let me write it down. Vital capacity. Okay. Now that we know the equation, Before we go through the answers, let us take a quick look at what all these terms are. Okay, so first, what is tidal volume? Tidal volume is the normal amount of air that we usually inhale and exhale while breathing. Right, so tidal volume is the amount of air that I am using while I am talking, right, while you are listening. That's the tidal volume, right, the amount of air you usually breathe in and breathe out. Then we have residual volume. Okay, before we go into residual volume, let's take a look at inspiratory reserve volume and expiratory reserve volume. So what is inspiratory reserve volume? Now, inspiratory reserve volume is the extra amount of air that we can inhale after tidal volume. Okay, so this is what we call the deep breathing, right? The maximum air that we can inhale into the lungs. So that's what expiratory reserve volume is. Then we have, inspiratory reserve volume is. Then we have expiratory reserve volume, which is the extra amount of air that we can exhale out of the lungs, right? After tidal volume, there is some air remaining in the lungs. So exhaling that air is called the expiratory reserve volume. Now let's come back to what residual volume is. Okay, now after expiratory reserve volume, after exhaling all the air from the lungs, even after that, there's a certain amount of air that is present in the lungs, right? So this certain amount of air that we can exhale out is the residual volume. Now, residual volume is really important for the body. Why? Because without the residual volume, the lungs and the alveoli will all start to collapse. So that's what residual volume is. Now let's go through the answers. Okay. What's the tidal volume? So the tidal volume is 500, right? And inspiratory reserve volume 3100 and expiratory reserve volume is 1100. Okay. And the vital capacity is 4700. The correct answer is the fourth one. Okay. Now let's go through question 23. Right? Parasympathetic nervous division of the autonomic nervous system of man. We know autonomic nervous system is the part of the nervous control that usually take care of the unconscious activities of the body, right? Such as the digestion, breathing. These are all controlled by the autonomic nervous system. We don't have voluntary control of them. Now, autonomic nervous system is divided into two parts. We have the sympathetic nervous system and we have the parasympathetic nervous system. Now, sympathetic nervous system is activated during flight or fight response, right? When our body is in danger, when we are stressed, when we are exercising. So during all these times, sympathetic nervous system is activated. Parasympathetic nervous system is activated when we are not in danger, during resting and digestion. Right? We also call this the resting and digestion response, because parasympathetic nervous division usually stimulates digestion. Right? Okay. Now let's go through all the answers. First, we have inhibit saliva secretion. Okay. We know saliva is one of the most important things during digestion, because saliva digests the food in the mouth. Right? So this is a activity of the parasympathetic division. So here they are saying inhibit saliva secretion, but during parasympathetic division, saliva secretion is stimulated, not inhibited. Again, we can eliminate that answer. Second one, dilates the pupil of the eye. Now, pupils are dilated only when we are in danger, right? Because the sympathetic nervous system is activated. When there are a lot of light coming into eyes, pupils are dilated. Again, when we are stressed. So, pupil is constricted during parasympathetic nervous system activation, not dilated. Again, that is the wrong answer. Third, we have relaxes bronchi in lungs. Okay, we know parasympathetic nervous division means relaxing, right? But we hear the word relax in bronchi, we think it is the right answer. No, it doesn't work that way, because when bronchi is relaxed, more air can enter into the lungs. Now, what happens when more air enter into the lungs? More oxygen is inhaled into the lungs. More oxygen, more energy is produced, and this occurs during sympathetic nervous activity, not during parasympathetic. Again, that statement is wrong. Then fourth one, we have stimulates the release of glucose from the from liver. Now, when is glucose released from the liver? Right, we know glucose is stored as glycogen in the liver. Glycogen is breakdown during high energy activities. High energy activities occur during sympathetic nervous division activation, right? Again, that statement is also wrong because parasympathetic division usually stores glucose as glycogen. Then fifth one, stimulates gallbladder. As I told earlier, parasympathetic division stimulates digestion, right? Now, if it stimulates digestion, it stimulates gallbladder. Why? Because gallbladder secretes bile. Bile, what the bile does? Bile emulsify the fat. So it is involved in digestion, right? So fifth one is the correct answer. Okay. Now let's go through the 24th question. Select the correct statement regarding human vision. First statement, they are saying changing refractive power of cornea facilitates binocular vision. Okay. We know light is refracted mainly in two places in the eye, that is the cornea and the lens. So in the cornea, the refractive power is constant, but in the lens, the refractive power is changing. Due to accommodation that takes place, the lens can be rounded, the lens can be more thin, right? So for this reason, lens have the changing refractive power, not the cornea. So first part of the statement is wrong. Changing refractive power of cornea, no, it is the lens. Cornea doesn't have a changing refractive power, and they are saying facilitates binocular vision. So the binocular vision, it means the way our eyes, right, from the left eye we see as one image, from the right eye we see another image. The way our brain combines both images, merges both images into one 3D image. So that's what binocular vision is. So this occurs in the brain and is facilitated by the movement of the eye muscles and the integration that occurs in the brain. That statement is also wrong. Second one, convergence occurs during distant vision. Okay. What is convergence? Convergence is the movement of the eye muscles towards the nearby object, and it is really important during near vision, not distant vision. So if you don't understand convergence, you can see now I am moving my finger towards my eye, and you can see the eye muscles moving towards the finger. So that's what convergence is, right? Again, that statement is also wrong because convergence is important during occurs during near vision. Third one, accommodation is important for near vision. Yes, this is correct. Why? Because accommodation is the changes that occur in the eye to see a nearby object. So what are these changes? It is the contraction of the ciliary muscles, loosening of the suspensory ligaments, and the rounding of the lens. What happens when lens are more rounded? More light is refracted towards the retina. So image is focused on one place and we form a very clear image. So that's what accommodation does, and that is important for near vision. Again, third statement is correct. Okay, fourth statement, photospin in rods provide night vision. We know there are two types of pigments in the eye. We have photopsin and we have rhodopsin. Photopsin is usually found in cones, not in rods. Again, that statement is wrong. And the pigment that is found in rods are rhodopsin, not photopsin, right? So rhodopsin in rods provides night vision. Photopsin are usually involved in the detection of color light, mainly red, green, and blue colors. Correct perception of visual objects occur in the frontal lobe of the cerebrum. No. Perception of the visual objects occur in the occipital lobe of the cerebrum. Behind the eye we have the retina. Right? Through the retina, the information goes into the optic nerve. So optic nerve carries the electrical signal into the occipital lobe where the image is integrated and correct perception occurs in the correct perception occurs there. Yes. Okay, now let's go through question 25. Which of the following combinations correctly matches the hormone and its function? First, we have ACTH. There's an ACTH, which is adrenocorticotropic hormone, stimulates adrenaline secretion. We know ACTH stimulates the adrenal cortex, right? Adrenal cortex, what they do? They release glucocorticoids and mineralocorticoids. And adrenaline is neither a glucocorticoid or a mineralocorticoid. And adrenaline is released by adrenal medulla, not by adrenal cortex. That statement is wrong. Then second statement, oxytocin stimulates milk production. Oxytocin doesn't stimulate milk production, it stimulates milk secretion or milk release in other words. But what is the hormone that stimulates milk production? It is prolactin, right? Again, that statement is also wrong. Then third statement, calcitonin promotes high calcium levels in the blood. Now, calcitonin doesn't promote calcium levels in the blood. Calcitonin actually reduces the calcium level in the blood. How are they going to do this? So they reduce reabsorption of calcium in the kidney, and they also increase absorption of calcium by the bones. In this way, calcium is reduced in the blood. That statement is also wrong. And fourth one, we have melatonin. Just melatonin increases the basal metabolic rate. Whenever it comes to basal metabolic rate, always remember it is the thyroid hormones, right? Specifically, thyroxine and triiodothyamine. Okay, again, that statement is also wrong. But anyways, what is the function of melatonin? Melatonin usually controls the circadian and the diurnal rhythm. In other words, they control the sleep and wake cycle. Always remember that. And fifth one, we have cholecystokinin. Okay, cholecystokinin triggers the release of pancreatic amylase. Okay, this is correct. Why? Because cholecystokinin is a hormone that is secreted by the small intestine. Cholecystokinin, what they do, they trigger the release of pancreatic juice from the pancreas. Now, this pancreatic juice, they contain a lot of hormones, and pancreatic amylase is one of those hormones in the pancreatic juice. That statement is correct. Okay. Now let's go through the 26th question. Select the correct statement regarding spermatogenesis of men. Okay, what is spermatogenesis? Spermatogenesis is the development of the sperm cell inside the testis, right? So first statement they are saying, spermatogenesis starts at birth and occurs throughout life. Spermatogenesis doesn't start at birth. Spermatogenesis starts only at puberty, but occurs throughout life, which is correct, but it only starts at puberty. Why? Because hormones that are necessary for spermatogenesis to take place is only released during puberty. So what are these hormones? So these hormones are the FSH and LH. So we need these hormones during spermatogenesis, and they are only released during puberty. Always remember that. That statement is wrong. Then second statement, spermatogenesis is promoted by testosterone, correct, secreted by Sertoli cells. Okay, here is the problem. Testosterone is not secreted by the Sertoli cells. Testosterone is secreted by the Leydig cell. We know in the seminiferous tubules we have two important kind of cells. We have the Sertoli cells and we have the Leydig cells, and Leydig cells are the one that releases testosterone, right? Okay. Then the third statement, primary spermatocytes are formed by spermatogonia through mitotic division. Yes. What are one of the initial cells of spermatogenesis are spermatogonia, right? Spermatogonia, they undergo mitosis to form primary spermatocytes. So this statement is correct. But the primary spermatocytes are the cells that undergoes meiosis, right? They undergo meiosis and form secondary spermatocyte. Okay, this statement is correct because primary spermatocytes are formed by spermatogonia through mitotic division. Fourth statement, Leydig cells provide attachment for cells that are in different stages of spermatogenesis. No, this statement is wrong. Why? They are not the Leydig cells. Usually, it is the cells that are present inside the testis, which is the Sertoli cells. The Sertoli cells, they provide nourishment attachment for all the different kind of cells in spermatogenesis, like the primary spermatocyte, secondary spermatocyte, the spermatids, the spermatozoa. All these cells are nourished by the Sertoli cells. Finally, we have all cells that undergo spermatogenesis are diploid except sperm cell. Okay, again this statement is wrong. Why? Because we know primary spermatocyte, yes, they are diploid, but secondary spermatocyte are haploid number of chromosomes. Again, secondary spermatids have applied number of chromosomes. Then again, spermatids also have applied number of chromosomes, right? Then sperm cells have applied number of chromosomes. But here they are saying only sperm cells have applied number of chromosomes. So which statement is wrong, right? Because again, remember what? Sperm cells, secondary spermatocyte, and the spermatids are not diploid, they are haploid. Okay. Now let's go through question 27. Which of the following statements regarding human development is correct? Okay, first statement, during fertilization, a sperm enters the mature ovum, penetrating surrounding epithelial cells. Interesting, right? Now we have to go back to what? Oogenesis. What we learned in oogenesis? In oogenesis, we have learned that the secondary oocyte is arrested at metaphase and they do not form the mature ovum until the sperm penetrates inside the egg cell, right? So it is in the secondary oocyte stage, not at the mature ovum stage. So the statement is saying sperm enters the mature ovum, penetrating surrounding epithelial cells. It's not a mature ovum, it is the secondary oocyte. That statement is also wrong. Then we have blastocyst reaches the uterus 3 to 4 days after fertilization. Now, what happens during fertilization? An egg cell fuses with a sperm cell and forms a zygote. Now, this zygote is a single cell, right? Now, this zygote is a single cell and they undergo mitosis and they form 2 cells, right? And these 2 cells become 4 cells and 4 cells become 8. So, in this way, they start to divide mitotically. So, during this stage, what happens? The egg cell, right? Egg cells start moving through the fallopian tube into the uterus, right? So when it's starting to reach the uterus, the zygote is in the morula stage, not the blastocyst stage, right? So morula stage, they have like 16 and 16 plus cells, right? So in that stage, they are in the morula, not blastocyst. So again, that statement is also wrong. Third statement, secretion of endometrial glands provide nutrition to the early embryo. Let's go in order. As I said, they are in the morula stage after 3 to 4 days, but after like 5 to 6 days, they become the blastocyst. Now this blastocyst, they go and get implanted into the endometrium, right? Now they are implanted. Do they have the placenta yet? No. Placenta is normally the structure that usually provides nutrition to the embryo. But at this stage, at the earlier stage, the placenta is not present. So endometrium secretes nutrition through the endometrial glands into the early embryo. Again, that statement is correct, right? Okay. Fourth statement, they are saying placenta contains only fetal blood vessels. We know placenta contains both fetal and the maternal blood vessels. If not, they won't be provided with the nutrition and the oxygen. The fifth statement, the heart of the fetus begins to beat by 8 to 10 weeks of pregnancy. Again, this statement is wrong. Why? Because heart is one of the first organs to be functional and it starts to beat about like 4 to 5 weeks, not 8 to 10 weeks. That statement is wrong. But we can only detect that heartbeat after 8 to 10 weeks, right? The correct statement is the third one. Now let's go through the 28th question. Which of the following statements regarding axial skeleton of man is correct? Now, I have added a lot of images for this question because it's really easy explaining the bones when there are images of the bones, Now, first statement, they are saying 3 pairs of ribs articulate with the sternum indirectly. What? 3 pairs of ribs articulate with the sternum indirectly. Here you can see. So this is the image of the sternum, right? This green color part. This is the sternum, right? Now we have 7 vertebrae directly attached to the sternum. You can see through costal cartilage, the yellow color part. 7 sternum are directly connected. But if you take a good look at the 9th, 8th, 9th and 10th ribs, you can see that they are not directly connected to the sternum, right? 8th vertebrae is connected to the 7th vertebrae and the 9th vertebrae is connected to the 8th vertebrae. But they are articulated with the sternum, but not directly, but indirectly. And even the 10th vertebrae, they are connected to the 9th vertebrae, even though the image doesn't show it. So that's the correct answer because 3 vertebrae, they are not connected directly to the sternum, but indirectly through other vertebrae on other above vertebrae. Right? And that statement is correct. Anyways, let's go through all the other 4 statements because all these other statements are really important, So, second statement, they are saying zygomatic arch provides surface for muscle attachment for the movement of the upper jaw. Now, a lot of students have a confusion of what this zygomatic arch is. We know the zygomatic bone, but what is the zygomatic arch? So, here you can see this is the zygomatic bone, right? Here you can see this is the zygomatic bone and this is the temporal bone. Now, the bridge that formed using the both zygomatic bone and the temporal bone is the zygomatic arch, right? Here, this is the zygomatic arch. So, they are saying in the question, the answer, zygomatic arch provides surface for muscle attachment for the upper jaw. So, this is the upper jaw, the maxilla. So, zygomatic arch is here, maxilla is here. It has nothing to do with the zygomatic arch. Nothing to do with the maxilla. So, zygomatic arch connects with the mandible, provides surface for muscle attachment for the mandible, which is the lower jaw, not the upper jaw. So, you can see here is the mandible. It provides muscle attachment to the zygomatic arch. Okay, then we have third one. Sacrum is formed from 7 fused rudimentary vertebrae. Okay, sacrum is formed not from 7 fused rudimentary vertebrae but by 5 fused rudimentary vertebrae. Here you can see 1, 2, 3, 4, 5 vertebrae that are fused together, not 7. Again, that statement is also wrong. Fourth one, sinuses are located in the nasal and the temporal bone. So, what are sinuses? So, sinuses are air-filled cavities that are found within the bone, right? So, they are saying it's found in the nasal and the temporal bone, but sinuses are usually found in the frontal bone, ethmoid bone, sphenoid, and the maxillary bone. Here you can see the colored regions are the sinuses that are present in between different bones, right? Again, there is no a temporal sinus or a nasal sinus. Okay. Final statement, until the development of the lumbar curvature, child cannot hold the head upright. Okay, holding of the child's head is by the cervical curvature, not by the lumbar curvature. Again, that statement is also wrong. Okay. Now let's go through question 29. Excluding patella, the number of bones in the lower limb of the human body is. Now, patella, what is patella? Patella is the knee joint, right? They ask except the knee joint, the number of bones in the lower limb. So let's start counting. First, we have the femur, which is the thigh bone. Then we have the fibula and the tibia, which is present in the calf. So which means 1, 2, and 3 bones, right? Then now let's count all the other bones that are present in the toes, right? Here we have 7 tarsal bones, right? 1, 2, 3, 4, 5, 6, 7 tarsal bones. 5 metatarsal bones, 1, 2, 3. So you can see this beautiful pink color, 1, 2, 3, 4, 5 metatarsal bones, and we have 14 phalanges, right? See 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. So 14 plus 5, 19, 19 plus 6, 20, 9 plus 7, 26, 27, 20, yes, 26, plus the femur, plus the fibula, plus the tibia, which means 29 bones, right? Okay. Okay, now let's go through question 30. Okay, now let's go through question 30. If 2 individuals having the genotype, okay, one individual, so this individual has 2 traits, right? One denoted by capital A and simple A, and another trait denoted by capital B and simple B. So they are saying for 2 particular traits are crossed. Okay, so the male also have this genotype, the female also have the same genotype, and they are crossed. They ask how many different genotypes can appear in the progeny according to Mendel's law. Now, there are 2 traits here, right? We can do a dihybrid cross. We can do one cross separately for the trait A, another cross separately for the one that is denoted by the letter B. Right? So male and the female both have the same genotype. So let's do for trait A, then separately do for trait B. And let's see how many different genotypes you could get for each trait. Right? Okay. Here we can get a homozygous dominant genotype and 2 heterozygous dominant. And finally, we can get 1 homozygous recessive. How many different genotypes we can get? Okay. What are the total? How many total genotypes? We can get 4 total genotypes, right? Total genotype 4. Different genotype only 3. Why? Because we have 2 heterozygous dominant genotypes. So different genotype there are 3. Now let's do the cross for this. So we can get again another homozygous dominant, 2 heterozygous dominant, right? Finally, we can get 1 homozygous recessive. Again here the total genotype we can get 4. Different genotype we can get 3. Why 3? Because there are 2 heterozygous dominant. Now for trait A we can get 3 different genotypes, and for trait B we can get 3 different genotypes. Now how can we get the total number of genotypes? We can get the total number of genotypes using the multiplication rule of Mendel's law. How? We multiply both these genotypes, then we will get the total number of different genotypes. So 3 into 3 is 9. Again you can see in the answers we don't have the answer that has 9, right? Again you will get free marks for this. I mean, people who did this paper got free marks for this because the right answer is not there. And let's go through question 31. Now, again a similar question. If a woman homozygous for blood group B marries a man who is heterozygous for blood group A, possible blood groups of their children would be. Now, the female has B blood group but homozygous. The male has A blood group but heterozygous. Why? Because they have a allele and a O allele. Now, this O allele is always recessive, right? So now let's do the cross. So here we can get 1 AB blood group. Okay, what happened? Okay, 1 AB blood group. Then we can get another AB blood group. Then 1 B blood group and another B blood group. Okay, here so we can get 2 AB blood groups and 2 B blood groups. Why? Because the O alleles are recessive. So the correct answer is the 4th one. Now let's go through question 32. Which of the following human genetic disorders are caused by gene mutations? So what are gene mutations? Gene mutations are mutations that occur in the nucleotides of the DNA. So we have 4 genetic disorders. First one, Down syndrome. Is Down syndrome caused by gene mutation? No. Down syndrome is caused by a chromosome mutation. That's why we call the Down syndrome trisomy 21. Why? Because a person with Down syndrome has an extra chromosome on the extra chromosome of chromosome 21. Usually a person has 46 chromosomes, right? But a person with Down syndrome has 47 chromosomes because they have an extra chromosome of chromosome 21. Again, that is a chromosome mutation, not gene mutation. Then second one, we have color blindness. Now color blindness is a gene mutation. This is correct. Why? It is caused by a gene mutation in the gene that encodes for the photopsin pigment. So photopsin is the pigment that is present in the eye that usually detects red, blue and green colors. And due to mutation in this gene, the person cannot detect these colors and it causes color blindness. That statement is correct. And third one, we have Turner syndrome. Again, Turner syndrome is kind of like Down syndrome but caused by chromosome mutation in the X chromosome. So this is called monosomy, not trisomy. Trisomy means they have an extra chromosome. If a person has 47 chromosomes, you can call it a trisomy. If a person has 45 chromosomes, we have a we call it monosomy. So Turner syndrome is caused by a monosomy of the X chromosome. So a normal person has 46 chromosomes, as I told, but person in Turner syndrome has 45 chromosomes and they don't have one X chromosome. And these normally occurs in women. So women have two X chromosomes, but the person in Turner syndrome only has one X chromosome. Then finally we have sickle cell anemia. Again, sickle cell anemia is also caused by gene mutation. How? So mutation occurs in the part of the red blood cells that code for the red blood cell, which causes the red blood cell to be sickle shaped. Right? So that is a gene mutation too. And the correct answer is B and D, which is the fourth one. Now let's go through question 32. 33, yes. Major steps in the isolation of DNA are as follows. Okay, we know why isolation of DNA occurs, because we want to extract the DNA from what? Maybe from an animal cell, from a plant cell, maybe a bacteria. Right? So these process is done to extract the DNA from all these cells, and these are done in a process, right? So this process goes in an order, but they have jumbled the order. We have to find the right order of the process. Now, first what happens, right? First, during isolation of DNA is present in the nucleus, which is surrounded by the nuclear membrane. Then on the top of that we have the cell membrane. So all these membranes should be broken down. For that, cell lysis should occur. So that's the first step, which is homogenization. And during homogenization, cell lysis occurs, which means we break down the cell, we disturb the cell membranes, the nuclear membranes, so that the nucleus is exposed to the solution. So the first step is homogenization, right? So that's the first step. Then so what's the second step? Now we have exposed the DNA, but now we have a problem. There's this enzyme called DNase that is present in the cell, that usually degrades the DNA if the DNA is exposed to the environment, right? So we have to inhibit this DNase. Why? Because we want to extract the DNA. So second step is the inhibition of DNase. Okay. Yes. What's the third step? Now we have inhibited the DNase. Now we have another problem. DNA is not freely floating, right? DNA is wound around histone bases forming chromatin. Right? Usually in the nucleus, DNA exists as chromatin. They are wound around histone bases or histone proteins. Now what we have to do? We have to dissociate this nuclear protein complex. So the nuclear protein complex is the nucleus, and the histone base proteins are called the nuclear protein complex. Now we have to remove the histone base from the DNA, and that is the third step. Right. Now what is the fourth step? Now that we have solved three problems, now we have another one single problem. What is that? Now this DNA is contaminated with a lot of things, right? Because in the cell we have RNAs and other molecules. So DNA is present between all these molecules. So first, then what we have to do? Then we have to remove all these contaminants. So that's what the fourth step is: removal of contaminants. Right. Finally, we can precipitate the DNA using ethanol. Now, first one is EDBCA. EDBCA, it is the fifth answer. How do I highlight it? Okay. Yes. Now let's go through question 34. When a migratory bird flies northward from Sri Lanka along a straight line path, the biomes it could encounter in correct sequence are. Now, this question is from this map. Like while I was doing A levels, I never went through this map. I thought this is not important, and they gave it for aesthetic purposes to make the environmental biology book look nice, right? Because nothing is nice in that environmental biology book. So anyways, they have given a question from this map, and let's answer that. Okay, they're asking, when a migratory bird flies northward from Sri Lanka along a straight line, all the biomes it could encounter. Okay, now let's draw a straight line from Sri Lanka. I'm drawing straight line from Sri Lanka. Now let's see all the biomes we could encounter. So the Sri Lanka is filled with tropical forest, so that's the first one, tropical forest. Then as we go above, we have the light yellow color part, which is the desert, right? So that's the second one. Then, as we go above, in the Russian part, we have temperate grassland. Again, on the north part of Russia, we have the coniferous forest, which is highlighted by light blue. Then finally, on the Arctic region, we have tundra, which is the fifth one. Now, tropical forest, desert, temperate grassland, coniferous forest, and tundra. So it's the second answer: tropical forest, desert, temperate grassland, northern coniferous forest, and tundra. Okay. Now let's go through question 35. Okay, now let's go through question 35. Two invasive alien organisms in Sri Lanka are. So what are invasive alien organisms? Invasive alien organisms are plants or animals that are introduced from another foreign environment to the native biodiversity, and these organisms, plants or animals, cause harm to the organisms surrounding them. Right? So these are what invasive alien organisms are. First, we have giant African land snail and citronella grass. So giant African land snail, it is an invasive species, right? Okay. But citronella grass is an indigenous species and they are not invasive, right? And they are usually found in savanna. And tilapia is an exotic species, which means they are from a foreign place, right? From a foreign environment, we introduce them to the biodiversity, but they are not invasive. So again, exotic species, but not invasive. Right, then we have tussock grass. Tussock grass is also another type of grass found in the wetlands of Sri Lanka. Okay. Then we have Guinea grass and Kogon grass. Guinea grass is a invasive plant species, and Kogon grass is usually another plant species that is found in the savanna grasslands. Again, first one is invasive, and second, Kogon grass is usually found in savanna. Again, that is not invasive. Then again, we have Guinea andara and Tamada. Guinea andara is a naturally occurring plant species in the tropical thorn scrubs, and Tamada is found in Dipatana. Yes. Again, both of them are not invasive. They are naturally found in certain places. Okay. Finally, we have lantana and water hyacinth. Okay, we know lantana and water hyacinth are both invasive alien organisms, right? If you know water hyacinth, they are present on the surface of the water, right? If you see certain ponds and lakes, you can see water hyacinth, and they really block the sunlight, kill the organisms that are present inside the surface of the water, because they don't they block the sunlight. The organisms that are present inside the water cannot do photosynthesis, right? For this reason, they are invasive alien organisms. Again, lantana is a very beautiful flower, but again harmful to the other plants surrounding them. So both of them are invasive plants. Now, fifth one is the correct answer. Now let's go through question 36. Okay, which of the following statements regarding viroids and prions are correct? Okay, viroids are the smallest infectious particles, right? So they only have the RNA structure. So this is how a prion will usually, viroid will usually look. And they don't have a protein coat or nothing. So they are just a RNA molecule. Okay. So first one, prions. Now let's viroids. Now what are prions? Prions are just misfolded proteins, right? They are not living. They are not a microorganism. They are just misfolded proteins that can cause different issues in the human body. How? Because these proteins go and affect the healthy proteins present in the body and also cause them to misfold. It causes them to become abnormal prions. So that's what prions are. Then first time we are saying Creutzfeldt-Jakob disease is a human disease caused by prions. Yes, which is correct. Prions usually affect cattle and sheep, but one of the diseases that is caused in humans is the Creutzfeldt-Jakob disease. This statement is correct. And second statement says viroids carry signal for their multiplication in host cells. So this is the structure of the viroid. This is just a RNA molecule, right? And the only signal they carry is for their multiplication. They don't carry anything else. Okay, then this statement is also correct. Now, third statement, virus have a short piece of, sorry, it should be DNA. I have put ONA. Okay, DNA protected by a protein coat. Again, virus doesn't have a DNA, they have a RNA, and they don't have a protein coat surrounding it. And you can see it is a naked piece of RNA molecule. Fourth answer, they are saying nucleic acids in prions replicate with the help of host genes. As I told earlier, prions are just proteins, right? They are misfolded proteins and they don't have nucleic acids, right? Again, that statement is also wrong. The correct statement is A and B, which is the first answer. Now let's go through question 37. Which of the following statements regarding endotoxins and exotoxins produced by pathogenic bacteria is correct? Okay, what are endotoxins and what are exotoxins? So endotoxins are toxins that are present in the surface of the outer membrane of the gram-negative bacteria. Always remember that endotoxins are always present in gram-negative bacteria on the outer membrane of it. So endotoxins are usually lipopolysaccharides, right? Lipid molecule with a carbohydrate component. Okay, then what are exotoxins? Exotoxins are substances that are secreted by both gram-negative and gram-positive bacteria, and when they are released into the body, into the host cell, they can damage the host cells in different ways, and even a very small amount of exotoxin is really lethal to the body. So first statement, they are saying both endotoxins and exotoxins are inactivated by heat. Now, endotoxins cannot be inactivated by heat because endotoxins are thermostable. But exotoxins are thermolabile, which means they can be inactivated by heat. So this statement is wrong. Now let's go through question statement 2. Endotoxins are proteins or lipopolysaccharides produced by gram-positive bacteria. As I told, endotoxins are only present in gram-negative bacteria on the surface of the outer membrane, right? Okay, you can remember this easily. How? Because gram-positive bacteria doesn't have outer membrane. So you should have outer membrane to have endotoxins. For that reason, gram-positive bacteria doesn't have endotoxins. Again, endotoxins are lipopolysaccharides, not proteins. Again, that statement is also wrong. Then third, we have exotoxins are produced by both gram-negative and gram-positive bacteria. Yes, this statement is correct because unlike endotoxins, exotoxins can be produced by both gram-negative and gram-positive bacteria. Now, fourth statement, exotoxin produced by Corynebacterium diphtheria act as an enterotoxin. No. Exotoxin that is produced by Corynebacterium diphtheria acts as a cytotoxin, not as an enterotoxin. Enterotoxin affects the intestinal cells, but cytotoxin usually affects the host cell membranes. Again, that statement is also wrong. Now let's go through the fifth statement. Endotoxins produced by different bacterial species cause different symptoms. We know endotoxins contains lipopolysaccharide. When the cells die, this lipopolysaccharide is released. So this lipopolysaccharide contains a specific component, right? So this specific component triggers the same set of symptoms, not different set of symptoms. Again, that statement is also wrong. Now let's go through question 38. Which of the following statements regarding metabolic products of microorganisms is correct? Okay. First, there are Saccharomyces cerevisiae used for the commercial production of invertase. We know invertase is an enzyme and it is produced using Saccharomyces cerevisiae. That statement is correct. Second statement, they are saying citric acid is produced through fermentation of molasses by Aspergillus oryzae. So we know for fermentation of molasses means fermentation of sucrose. Right? There's no Aspergillus oryzae, but citric acid is produced through Aspergillus niger, right? Not oryzae, it's Aspergillus Niger. Okay, then that statement is wrong. Then third one, riboflavin, which is vitamin B2, is produced through fermentation by Bacillus subtilis. Riboflavin is produced by fermentation by fungi. Bacillus subtilis is usually involved in the fermentation to produce amylase. Okay, always remember that. Bacillus subtilis, amylase. Riboflavin is produced by fermentation by fungi. Now let's go through the fourth statement. Tetracycline is a secondary metabolite produced by Streptomyces graecosus. Now, what is a secondary metabolite? Okay, secondary metabolites are substances that are produced by microorganisms that are not essential for their survival but gives them an advantage, right? Now, for example, certain bacteria, they produce antibiotics. Very ironic to hear. Why? Why would the bacteria produce antibiotics? Because to reduce the competition of other bacteria. So that's what secondary metabolites are. They are not essential, but they are produced by certain microorganisms for their advantage. So there's a tetracycline is a secondary metabolite produced by Streptomyces graecosus. Again, this statement is wrong. Why? Because tetracycline is produced by Streptomyces aurofaciens, not graecosus. Again, that is wrong. Last statement, ethanol is a secondary metabolite produced through fermentation of sugarcane sap by microorganisms. Now, is ethanol a secondary metabolite? Okay, secondary metabolite is not important for the survival of the organism, but they are produced only for an advantage. But ethanol is produced by anaerobic respiration of yeast, right? So anaerobic respiration is essential for the yeast survival. If it doesn't carry out anaerobic respiration, the yeast will not survive, it will die, right? Ethanol is not a secondary metabolite. For that reason, ethanol is a primary metabolite, right? They are formed as a result of anaerobic respiration. Again, that statement is also wrong. Now let's go through question 39. Which of the following may contribute to keep the immunity of ornamental fish in a home aquarium at a high level? Now, what are ornamental fishes? Ornamental fishes are the fishes that are used in aquariums, right? We use them for decoration, for beauty, right? And we don't usually eat those fishes. So they're asking how to keep the immunity of these fishes really high. So the first statement they are saying, using correct feeding regime has a biosecurity measure. Yes, if you feed them correctly, their immunity is high, but has a biosecurity measure. So biosecurity measure is to prevent them contaminating with other pathogens. So feeding them properly is not a biosecurity measure. That statement is wrong. Second one, replacing entire volume of water fortnightly. Anybody who is using an aquarium, who has a tank at home, they know they don't replace the entire volume of water. They replace only 25% of the aquarium water. Again, that statement is wrong. Even though it will keep the immunity high, if we change the water, if we maintain the water, we can keep the fish healthy. If the fish is healthy, the immunity is high, but we don't replace the entire volume of water, right? That statement is wrong. Turning off aeration at night. So what is turning off aeration? Turning off oxygen in the tank, right? They are saying turning off oxygen in the tank every night keeps the immunity high. Is it? Right? If we turn off oxygen every night, the fish will die, right? We don't want that. We only turn off aeration when you're cleaning the tank. In that statement is also wrong. Fourth one, keeping the lights of the aquarium switched on continuously. Now, what happens if we keep the lights on aquarium continuously on? The algae that is present in the aquarium, they get a lot of light, right? They get a lot of light and they undergo photosynthesis and produce a lot of algae, reproducing rapidly. We don't want that, right? So again, fourth statement is also wrong. It doesn't keep the immunity of the fish high. Fifth statement, maintaining correct stocking density of compatible plants and fishes. Now, what is maintaining correct stocking density? Maintaining correct stocking density means only having a certain amount of fishes and plants, right? Not overcrowding them. What happens when you overcrowd them? They don't have enough food, they don't have enough oxygen, they can't move, so the immunity will become very low. So that we should maintain a correct stocking density of compatible plants and fishes. So what are compatible plants and fishes? So compatible plants and fishes means fishes that can live with each other peacefully. They don't fight with each other. They don't fight for the food, right? So if a fish fight for the food too much, one fish will die, right? Because it doesn't get enough food. So compatible fishes, right? Fishes that can live with each other. Same with plants. So maintaining correct density of plants and animals will keep the immunity of the fish high. So that statement is correct. Now let's go through the 40th question. Which of the following statements regarding genome project are correct? So what is the genome project? Genome project was this huge project that was done for years, where they researched the genes of the humans, right? They researched every nucleotide that is present in the human. So that was the genome project. Identify all the proteins that are present in the human. So there's one of the aims of the Human Genome Project is to determine the sequence of 20,000 base pairs of human DNA. Do we have 20,000 base pairs? No, we have 3 billion base pairs. So the aim of the human was to determine the sequence of 3 billion base pairs and 20,000 what? 20,000 genes that are present in the human DNA. Again, that statement is wrong. B answer, Human Genome Project is yet to be completed. Again, that is wrong because Human Genome Project started at 1990 and it was completed on 2003. That statement is also wrong. And C answer, they are saying Human Genome Project has led to description of molecular activities of human cells. Now, Human Genome Project was done to identify the genes, right? When they identify the genes, there are genes that code for proteins. So when we identify the genes that code for proteins, we can understand and the functional and the structural activities of that protein. So this statement is correct. And last statement, genome project of Escherichia coli is already completed. Yes, genome project of Escherichia coli, the mouse, and even genome project of the rice is already completed. That statement is also correct. The correct answer is the C and D1. Okay. Whew. Now that we have completed the first 40 questions of the MCQ paper, on our next video we will be going through the last 10 questions in detail from the G.C.E level 2023 paper. Until then, see you guys. Thank you.